now, all the COVID-19 coronavirus deaths are rising. Guess what's dropping massively? Deaths from pneumonia, deaths from the flu, and deaths from heart disease and heart attacks. Don't you think it's a bit ironic? What, are people not dying from heart attacks anymore? Are people not dying from pneumonia? Are they not dying from the flu? Nope. Apparently COVID-19 and coronavirus fixed that. So now every person that dies now, for the most part, is getting labeled, toe tagged with COVID-19 coronavirus.
Physician, I received an email last week from the Department of Health. They were saying, you know, you don't have to have a confirmed laboratory test for COVID-19 in order to make the death certificate be COVID-19.
So, wait. Sir, I don't mean to interrupt you, but that and what you just said, I think is critically important. Can you repeat what you just said, please?
Well, last Friday, I received a 7-page document that sort of told me that if I had an 86-year-old patient that had pneumonia but was never tested for COVID-19, but sometime after she came down with pneumonia, we learned that she had been exposed to her son who had no symptoms, but later on was identified with COVID-19, that it would be appropriate to diagnose on the death certificate COVID-19.
Why would
So I'm going to change gears now a little bit and talk about something different, something that most of you have never heard of whatsoever, and it's called exosomes. And I'm going to talk about what they are and why they're relevant for this discussion. So exosomes are something that is naturally occurring in the body. So I have this diagram here, and if you look at the top right of the diagram, you'll see that this is a normal cell here outlined, and inside the cell you have these vesicles. And inside of our cells there are a number of different little organs, they call them organelles, and they're generally contained by a membrane. They come in different shapes, but lots of them are in this structure. It's essentially like a spherical blob that has the same membrane as is the outer membrane of the cell, a lipid bilayer membrane, and it can contain various types of chemicals. Now, this is a specific type of vesicle that will end up merging with the cell membrane at the surface under certain conditions and release these exosomes out into the extracellular fluid, and they'll get into the circulation and be distributed around the body. So while they're inside the cell, they're called MVEs, which stands for multivesicular endosomes. So in other words, they have a bunch of exosomes or vesicles inside them. And they release these on a regular basis, just day to day. And there are many things, though, that can induce this process and accelerate it and increase the number of exosomes that it releases outside of the cell. And so these exosomes leave the cell, and you see they have these little squares on them. And these little squares are like a lock. And what they do is they go around the body through the circulation, and they're looking for the right key to fit their lock. And that's called the target cell. And depending on what kind of cell releases them, they might have different keys and different locks, so they're targeted to different parts of the body. And these are mostly thought to involve communication. So communication between one cell and another cell, between one part of the body and another part of the body. And they can have many functions in this communication, which I'll get to momentarily. So here on the left, this is an electron micrograph, or a picture from an electron microscope of exosomes. And you can see here an exosome budding out of the cell. And it's essentially a spherical or circular, this is a cross section, so this is like a slice of the tissue, where on the periphery there are like these kind of globular densities or little dots or circles, right? And inside the cell here where it says MVB, sorry, these are the same as MVEs, that's where all these exosomes are when they're inside the cell before they bud out, okay? And now we have a picture on the right which allegedly shows this COVID-19 virus. And you can see that there are these vesicles budding out of the cell in a circular shape with these globular dots on the periphery. So essentially, it's the same thing. Now, you might think that this looks a little bit fuzzier on the exosomes and it looks a bit sharper in this picture. And I want to tell you that the reason for that is because when you're cutting these thin sections of tissue to make these slides, you're using this device called a microtome. It's like a vibrating razor blade. And it's technically difficult, and sometimes the tissue doesn't cooperate well, or sometimes the person is less skilled, and you might not have a perfect slice. On this COVID-19 slide, this is, I've not seen a better slice than this. It's absolutely perfect. The one on the left is a little bit thicker, and so that's why it looks a bit fuzzier and less sharp, but essentially you're seeing the same exact thing. And I want to take a look at the same comparison when we see both of them inside of the host cell. Okay, so on the bottom left here, this is a COVID-19 cell. And you see right here is supposedly this is the virus particle inside the cell. And once again, you see a circular shape. And inside, you see these round globular aggregates. Okay, now look at the top right. And this here is showing you this is less magnification, and this is actually a nerve cell. And this is the MVE. So these are a bunch of exosomes inside here. And you see a circular structure filled with these globular particles. So once again, same thing, also same size, both about 500 nanometers in diameter. Okay, you see the scale lines here, so you can see for yourself. So now we have a series of 2 comparison photographs under electron microscope of the virus and an exosome outside the cell, and a virus and an MVE inside the cell, and you can see that they are identical in appearance. So let's look at actually some physical parameters of these and continue our comparison. So I've mentioned a little bit that they are roughly the same size when they're inside the cell, same size when they're outside the cell. Now there is some variability here. I've looked at lots of these photographs, and I see in both cases there is some variability in the size. So these numbers are not precise, but in every case they are essentially compatible in their range of sizes. Now also the receptor, now this is a key thing. Now remember I mentioned that lock and key mechanism. Well, actually with the COVID-19 papers, they have discovered that on its surface it has a receptor for ACE2. Now ACE2 is an angiotensin converting enzyme. It's an enzyme in our bodies. One of its functions is that it works with the kidney to regulate blood pressure, and there are blood pressure medications that inhibit this enzyme. But in this paper, what they said is that this receptor is actually how the virus invades the cells. Now, I also was able to find a paper where they've identified exosomes coming from our own body that also use the ACE2 receptor as their lock and key mechanism to find their target cells. So exosomes and COVID-19, the same exact receptor on their surface targeting the same exact cells. Now, these both contain genetic material, and it's both in the form of RNA. No DNA, only RNA. And these structures are both found in the lung fluid. So the lung cancer test that I showed you earlier found exosomes in that fluid, and the lung fluid showed the COVID-19 as well. So you can see that the more we take out this comparison forward, we see that they're essentially the same in every important way. So I happen to be looking in the virology literature, and actually they also think exosomes and viruses are possibly the same thing. So this is Dr. James Hildreth, a very prominent researcher and academic physician in the field of virology and HIV research. He's currently the president and CEO of Meharry Medical College, but he was a full professor at Johns Hopkins, and he wrote this paper with 2 of his colleagues there. And what he said, and I quote, "The virus is fully an exosome in every sense of the word."
I have to also apologize that the hashtag end the lockdown was one of the top trending items on Twitter in Canada yesterday. That was partially, actually mostly probably because of Dan Dix, who also put out a complete fake news video saying that some people were questioning the lockdown, which is terrible. You should never, ever question the lockdown and what the government says. Never, and I mean never. Or what the media says, the mainstream media, or the pharmaceutical industrial complex, or Bill Gates.